Thank you, Jonathan, and good afternoon, everybody. And I want to thank the organizers for inviting me to participate in this important discussion. Today's topic of discussion is the participation of older adults in clinical trials. The hypothesis is that there is inadequate representation of older adults in clinical trials, even though in majority of clinical trials, the eligibility criteria includes all adult subjects over 18 years of age, and the upper limit of age is not specified in most trials. Why then older adults are not enrolled in clinical trials? The main reason is that often exclusion criteria in clinical trials include some comorbid conditions, and it is not uncommon that older adults have comorbid conditions. We have to also ask if there are other reasons for low enrollment, perhaps the accessibility of clinical sites, overburden of frequent hospital visits for protocol-specified testing, follow-up, etc., and not being able to tolerate toxicity of some drugs. For example, in cancer trials, often the protocol specifies for frequent assessment of tumor progression with radiological scans. Nevertheless, these older patients are treated in clinical practice, and therefore, we have to understand the risk-benefit of the new treatments in this population by finding ways to increase enrollment of older adults in clinical trials. What are some of the ways to increase enrollment in this population? Without compromising patient safety, we can perhaps expand eligibility criteria to include patients with some comorbid conditions. We can examine different clinical trial design options to ensure inclusion of older adults and address some of the roadblocks, for example, by choosing less burdensome outcome assessments. Another important aspect, we can examine different clinical design options to ensure inclusion of older adults and address some of the roadblocks, for example, by choosing less burdensome outcome assessments. Another important aspect is to educate patients and investigators that it is important for older adults to participate in clinical trials. Per the US Census Bureau report, 16.5% are estimated to be above 65 years in the overall US population. However, we all understand that proportion of older adult population varies with disease. Let us now look at the FDA experience in 2019. 48 products were approved for the first time in various diseases. The sample sizes of the studies that led to the approval of these 48 products ranged from as small as 56 to over 11,000 patients. In studies evaluating 40 products, participation of women varied from 10 to 91% of the population. 6 products were for the treatment of diseases in women only, and 1 for disease in men only. The participation of patients 65 years or older varied from 1% for the treatment of a rare blood disorder to 90% for treatment of age-related macular degeneration. This proves that there is no inherent reason that older adults cannot or will not participate in clinical trials. When the disease affects mainly older adults, the investigators and the pharma companies can enroll this population in clinical trials. 7 of the 48 products approved had less than 10% participation, and 30% had less than 30% participation by patients older than 65 years. There were no patients over 65 years in studies of 8 approved products. Further details on participation of 75 and older was not publicly available for majority of these products. So one thing we may want to consider is that we should be reporting the demographics much more clearly even for 75 years and older. Of note, participation of Black, African American, as also other minority racial groups were small. Because of my extensive experience in drug development in oncology, I will now present some of the statistics in cancer. Singh et al. Conducted a pooled analysis of 105 registrational studies spanning over 10 years on about 250,000 patients. Although in the general cancer patient population, 56% were estimated to be over 65 years, in the pooled data of the registrational studies, only 41% of patients enrolled were over 65 years. Similarly, only 12% of the enrolled population were over 75 years, although in the overall cancer population, 29% were expected to be in this age group. In 2019, 17 new molecular entities were approved for the treatment of oncologic and hematologic diseases, of which 9 had over 30% participation and 5 had less than 10% participation by patients 65 years or older. Here are the studies with less than 10% enrollment of patients 65 years or older that led to the approval of 5 oncology and hematology products. 2 of these are for the treatment of sickle cell anemia, where the expected average lifespan of patients is less than 50 years. 1 product is for the treatment of anemia in beta thalassemia patients, and the average normal lifespan of these patients are also not long. The other 2 approvals are in very rare diseases. Of note, 91% of patients enrolled in study evaluating crizanlizumab were Black or African American, as sickle cell disease affects this population more than others, suggesting that this population can also be enrolled adequately in clinical trials. Recently, Flora Mulkey at FDA looked at all randomized studies evaluating immune checkpoint inhibitors in various cancer types. In this analysis, there were 22 randomized studies with a pooled data on nearly 15,000 patients. Of these patients enrolled, 43% of patients were 65 years or older and 10% were 75 years or older. Of note, only 25% of patients enrolled in these studies were from US. Here are some points to consider to increase participation of older adult patients. Prevalence of disease in older age population, access to care facility, frequency of outcome assessment, whether outcome assessments are invasive or non-invasive, thoughtful consideration of exclusion criteria based on toxicity of the drug being evaluated, and any drug-drug interactions, as many older patients are likely to be treated for comorbidity. FDA has conducted public workshops on the topic of expanding eligibility criteria without compromising safety of patients, and 3 guidances have been published. One of them on enhancing the diversity of clinical trial populations, which includes examination of eligibility criteria, enrollment practices, and clinical trial designs.
Pardon me, Raji. Just giving you your 2 minutes. Thank you.
Sure. This guidance is not disease specific. 2 other guidances are specific to cancer, one of them specifically on inclusion of older adults in cancer clinical trials. I have listed these references at the end of my talk. In considering clinical trials, there are 3 ways by which we can learn about the use of investigational product in the older age population. During the early phase of drug development, one can add a small cohort of elderly patients to better understand safety and tolerability of the treatment. This can be sequentially considered after understanding safety in younger adults or more healthier adults. It's also important to understand any differential dose selection, modification, and drug-drug interaction in the older age population. This early study will also allow to evaluate feasibility of outcome assessments in this age group of patients. If the treatment is deemed safe and feasible, then both investigators and drug sponsors will be more willing and likely to enroll older patients into their studies. Secondly, in late phase or registrational studies, there are ways in which we can include the older patient population or potentially high-risk patients. And thirdly, data may be collected in post-marketing studies. In a RCT, generally a restrictive homogeneous population are enrolled so that if there is an effect of the treatment, it can be evaluated with minimum number of patients without any variability in the population. The more variability or heterogeneity, it's harder to show treatment effect. However, we can include the restricted homogeneous population and the older age high-risk population with the intention to conduct the primary evaluation of treatment effect based on the homogeneous restricted population. This can be accomplished by hierarchical testing in the modified intent to treat population first and then ITT population or the intention to treat population. There is also a proposition, we can also think of simultaneous randomized controlled trials in restricted population and single arm cohort in the high risk population. Of course, in the single arm, they are both analyzed separately, and the single arm high risk cohort, you can only provide descriptive statistics. This is my last slide. So the key considerations are who should be in the expanded older age high risk population that can be included in the clinical trials. So we have to consider what comorbidities they have, drug-drug interactions, and the level of toxicity that's acceptable in this patient group. In the randomized controlled trial, what proportion of patients in older high-risk group should be included? Should we limit the number of patients in this stratum? And the primary hypothesis of type 1 and type 2 errors, and if it's a time to event analysis, number of events for the final analysis, and the benefit risk assessment based on restricted population only. Is the hierarchical testing that I mentioned feasible? What if more events occur in the high-risk stratum and the total number of events are reached before you have sufficient numbers in the restricted population? Consideration of safety assessment in how do we evaluate safety in this high-risk population is also important. Single-arm cohort could consider enrolling patients only in certain sites, but it's difficult to interpret toxic events, in particular deaths, without a control arm in a single-arm study. Post-marketing studies, yes, can be done, but we have to have reproducible outcome definition and assessment and measures of safety evaluation consistency in recording data. Thank you.